Hello friends. So we will continue our coverage of indoor bioaerosols, specifically looking at the sources. So sources of bioaerosol particles in the indoor atmosphere. The common sources of indoor airborne biological pollutants include outdoor air, human bodies, wallpapers, carpets, suspended particles, air conditioners, also animal excreta. So there are a number of sources from which bioaerosols can be emitted to the air, and once they are in the air, then they can behave depending on the particle sizes. They may settle down or remain suspended, or may act as condensation nuclei and grow and remain suspended, perhaps in the accumulation mode, or if they are in bigger sizes, then they will settle down. So once they are emitted, and these are a number of sources, so we will have bioaerosols almost in all the indoor environments to different extents depending on what the sources are. These are the different aspects of the microbial contamination of air. First, we have the sources. So sources can be our building characteristics. Then if it is an old building or a new building, how moist it is, are there molds, are there bacterial growth in the bathrooms, or what is the situation in the kitchen. Then they may be sourced from dust, resuspension of dust, or outdoor soil coming in and being resuspended. We can have human contribution, so biological material either exhaled by human or released from the skin. Then pets, so pets excreta, pet dander, pet saliva, pet feces. Then indoor plants may release maybe pollens. Then outdoor and environmental determinants are also there. So humidity, temperature, moisture. Then these are the sources. These would relate to give us some microbial diversity, which could be different bacteria, fungi, and viruses. These may then pose different health risks. Microbial infection, maybe some can be cancerous, or respiratory illnesses, hypersensitivity, or exhibit toxicity. To understand these quantities, we will have different sampling strategies, which we will cover in a later week. We can have active sampling or passive sampling, and once these samples are collected, we can have our culture based methods and culture independent methods or non-culture based methods, and these will be covered in a later week. And once we have this analysis and understanding, we can undergo for mitigation measures, which would be basically routine monitoring. So by routine monitoring, we would know whether to undertake some mitigative measure, do some action, remove some spores, whether to do some renovation. So we need that monitoring. We can also change our ventilation characteristics. Maybe we want, if the indoor sources are high, maybe we can have more ventilation, so that the indoor sources, the emissions from indoor aerosols, the emissions of indoor bioaerosols are flushed out and fresh air is there. We can possibly install air cleaners and purifiers to remove the biological contamination. Also, we can have education and awareness to make people aware of the bioaerosol pollution, bioaerosol toxicity, and that will help them modify or undertake behavior that will minimize or reduce the risk to bioaerosol exposure and the subsequent health risk. So these are the different aspects again. So we will have human occupants in the different rooms that may influence the bioaerosol settings or bioaerosol loadings. Then people may be in the bedroom, and on the bed when we are moving we will shed some skin, and then that biological material can become resuspended. In biological room, in living room, we may be sitting on sofas, and again some skin may be released, skin fragments. Then related to water activity, because in a water damaged or moist setting, in a old house or a damp house, will have lead to some biological growth. Biological growth can also happen in the bathroom. Then in kitchen, where we may have old foods, this can happen. Then we have sources such as pets, which pet dander, pet saliva, pet feces, plants, then dust mites, and then other special sources are healthcare facilities, which may actually be also source of a lot of biological or human derived biological material, and possibly also a lot of infectious material. Then we may have the outdoor air influencing, so what is coming through doors and windows, and also what is the situation of the air conditioning system. Maybe that also can bring bacteria or microbes inside. So these bioaerosols can deteriorate indoor air quality. When we have humans, some of the bacteria or microbes can actually be normal, because we are anyways biological being and we have bacteria and microbes inside the body. So some of the things, like when we speak, of course microbes are coming out, but they may be normal. The issue becomes when a person is sick, and then for example sick from viral infection, and then the person can release the viruses. That will become a problem that relates to the poorer indoor air quality. In the bedroom, as we already seen, we can have bacteria, fungi and fungal spores. We can have this endotoxin. Then we can have the dust mite allergens. In the kitchen, because we have garbage bins, food and stale food, we can have bacteria emissions, we can have fungi also, we can have endotoxins also, and some of them could be basically related to cockroach allergens as well. In bathroom and toilets, we may have bacteria, fungi and endotoxins. Similarly, from pets also, we may have bacteria, endotoxins, the skin dander, and also various allergens. When we look at outdoors, can lead to indoor air quality deterioration in terms of bioaerosols, and this can be all bacteria, fungi and viruses. So when we have this generation of bioaerosols, let us say we have the susceptible host which is inhaling these propagation of virus particles. And where are they coming from? Direct generation, so person to person. So it could be cough or sneeze or a regular speech, and that may emit some bioaerosols, and breathing. So all this will lead to generation of bioaerosols which are now suspended in the air. There can be indirect generation, for example from toilet plumes. So that may emit a lot of bioaerosols which can be in the bathroom, and if the bathroom is open to a room, they can come in the room. Then in surgical and dental settings, a person's mouth is open, so there may be some emissions and contribution to the indoor bioaerosol, and the ventilation. So bioaerosols coming from outside to the inside, and all of this can again become part of the indoor bioaerosol setting. We can also have resuspension of previously settled bioaerosols, which may be part of dust or other particulate matter. So when we are walking on the floor, there may be resuspension happening. When we are performing activities such as vacuum cleaning, this may be happening. When we have protective clothing, maybe that protective clothing has bioaerosols. So here is an example of a mask. And if we do not dispose immediately and this is subjected to some abrasion or shear, we will have resuspension. And we may have exhaust from different sources, or maybe like air condition or the fans, they may resuspend the settled dust. And all this comes in the indoor bioaerosol setting now. And then this is then inhaled by a susceptible person who was previously not infected but now is inhaling all these bioaerosols. Human beings are generally considered to be a very important source of airborne bioaerosols because we have a host of microbes within our body. So human activities, which include talking, sneezing, coughing, which these activities basically bring out the microbes from our body to outside. And we have other activities that make suspension of bioaerosols better. For example, walking, which will resuspend the bioaerosol. Washing activity may lead to some bioaerosols. Toilet flushing, so it is human bacterial material which was out, but due to the flushing this will come out in suspension. So we are sort of resuspending the bioaerosol. So all these activities and possibly many more will further increase the airborne microbial load. Sneezing, out of all these, sneezing is perhaps one of the most vigorous mechanisms, especially when we are talking about coughing and talking. Sneezing is more vigorous, and it may generate a very high number of the order of millions of lower sized droplets, micron, sometimes submicron sizes. So these kind of then bioaerosols can possibly remain suspended for a long time. Therefore, anyone else in that area may be inhaling those kind of bioaerosols. This is another schematic of viral resuspension. So we will start from this part. So resuspension induced by air swirls from walking. So as the person is walking, we have these air swirls happening around the feet, which leads to the suspension. And maybe the same person is exposed to this suspended matter or a person behind. The other is resuspension induced by vibrational forces from moving of protective clothing. So if the person was wearing gloves, if the person was wearing mask or a protective gear, protective cloth, when they are moving these gloves, they may do it sometimes vigorously. Same for the mask, and when the person is taking out the cloth, there is some shaking. So vibrations, and that may release some aerosols to the air itself. The third type would be forces from vacuum cleaning. So when a person is moving the vacuum cleaner on the floor, some particles are sucked, but around the edges we will have some suspension of particles. as we move the vacuum cleaner. The next spread could be through the exchange at doorways. So this could be air coming in and out, or the resuspension of particles. When we open the door, there may be resuspension of dust, and the airflow would be such that we have the air coming in and it will take the resuspended particle in. Then we have the ventilation systems, in which the air vortices will induce the mixing of air with the infectious droplets. So on the air itself, and maybe due to which are going in the air from the resuspension. So this airflow from the ventilation system may also enhance or initiate resuspension. Then we may also have transmission of particles from another room due to these mechanical, wind forces, wind which is forced through fans. So these air vortices will of course emit the particle out, and depending on how the fans are, ceiling fans and the exhaust are, some of it may be resuspended and go to another room. Sneezing and coughing can generate a wide variety of virus-laden droplets of lot of different sizes. We may have sizes of more than 100 micrometer large drops, which will probably deposit within few seconds within 1 meter. Then we have the small droplets, which may possibly go 1 or several meters and remain suspended for maybe half an hour, tens of minutes. And these are these green particles, 5 micron size. And then we have the small droplet, this 1 micrometer, which may form condensation nuclei, or they will remain like this because they are in the accumulation mode. And these smaller droplets can remain, they are termed as droplet nuclei in this figure. Anyway, so these small droplets will remain suspended for up to 12 hours, or they may remain suspended, and their distance travel could also be like 40s, so tens of meters. So that means such kind of droplets can persist for a long time and also travel substantial distance as far as humans are concerned, because 40 meter means a lot of people can come within this 45 meter zone, and also the droplet is there for up to 12 hours. So these smaller droplets can be the causes of future infection. These small water droplets thus are responsible for these short range, long range routes and the indirect contact routes, because for example here, if we have the droplets, the larger droplets may be settling down, the medium droplets will be exhaled by someone else if the distance is not too large, and the shorter droplets will go for longer time, and this person is of course also inhaling the small droplets, and the small droplets can also go for longer distance and to another person, and also they may be attached to other surfaces in between or other people's clothes, and when they are vibrated, these aerosols will be suspended. When we come inside the house, toilet flushing is one of a very major cause of bioaerosol emission. This is a picture taken after flushing of the toilet, and these are the aerosols that are being emitted to the air. So as we can see, the toilet flushing has released or spewed aerosols to a substantial height. In fact, they can go several feet above the bowl after flushing. So when we flush, these bioaerosols are in the air, and then they become part of the bathroom air. And then anyone who uses the bathroom will be exposed to these bioaerosols. And this is important, especially in public bathrooms, where maybe there is a low exhaust, and if there is high occupancy, that means one can have bioaerosol exposure in such kind of settings. It has been found actually that bioaerosols from the washroom of the office buildings have been found to increase the lead to an higher indoor airborne bacteria burden, especially in the morning to evening hours when the occupancy load may be higher. So it has been shown to influence the indoor bacterial burden. As such, an increase in bioaerosol concentration was observed as the increase in occupants was observed. So more occupants were leading to possibly more washroom use, leading to more emissions of the bioaerosols to the air. Numerous kinds of bacteria live in different parts, inside and outside the human body. So about 10 to power 12 to 14 types of microbes may be living in the skin and in our digestive tract as well. So those microbes can go out either through the breathing or in the feces when they are situated in the digestive tract. Also, humans will shed bacteria-laden kill scales into the air. So parts from our skin may go into the air. Of course, very small sizes so that they are not visible to the skin, visible to the eye. But these emissions will happen. In fact, studies have found a positive correlation between human activity and the emission rates of total airborne particles. Of course, one cannot say when we are seeing a higher emission of airborne particles from human activities, one cannot say that everything is bacteria, or even if it is bacteria, everything is coming from within the body or the skin, just by measuring the aerosol count, bioaerosol count, because bioaerosols in an indoor environment can be from many different sources. However, if one to go and look for specific aerosols from the human body and then measure the same in the indoor air, and if a match is found, or look at microbes specific to the human body and then see how many of them are in the indoor air and to what extent. If there is a good match, then we can say that human body or human shredded microbes are the main contributor to the bioaerosol of an indoor air environment. So as I said, humans will release their own distinct bacterial communities, and these communities can be used to identify the individual occupants also in some times. So one, we can find if the bioaerosols are mainly contributed by humans, and within that, if one were to go into more intense research, it may be possible to even find the individual occupant who may be responsible for certain bioaerosols. It has been shown that the gender of the people occupying an indoor environment, the type of clothing they are wearing, what are the lipids on the skin surface, or use of creams or other things, or the property of the skin itself. These kind of things have been shown to influence the emission rate of bioaerosols from human. As just a reference, it has been found that seated office work resulted on an average emission rate of 0.9 million particles per person per hour in the office setting. So that is a huge number of particles, and this is actually always occurring. So it is not that all the bioaerosols are problematic, because we are always living in such kind of settings, in house or office or any public place. There are millions and millions of bioaerosol particles. The thing is that how many of them can be infected, and then a person has to inhale them and then actually get infected. So a lot of processes are there, but in general there will be a lot of bioaerosols in the air. When we have respiratory infections such as what we know COVID-19 or SARS and influenza, they primarily spread through virus bearing liquid particles, and these virus bearing liquid particles can be from the carrier's nose, so maybe while exhaling or by sneezing, and this can be emitted also from the mouth when we are breathing or coughing, and sneezing of course from the nose. So these kind of things can be primarily spread through the nose and the mouth. As such, in the enclosed environment, human beings can be important, or it is written largest. For some studies they have found to be largest artificial in the sense we are not using a bioaerosol generator, but we are maybe an important source. The take home is that human beings would be a very important source of bioaerosols in any kind of indoor enclosed setting. And then when humans are the main source or an important source, they will influence the structure and composition of the microbial flora within the environment, within that particular indoor environment. So sort of the microbial characteristics or microbial profile of the indoor air is profoundly dependent on or profoundly influenced by the presence of humans and what the humans are emitting. Now we go from human we go to the resuspension of sources. So all the sources leading to the suspension of dust particles and generation of airborne microorganisms, and these would be sweeping of floors, so broom sweeping or any kind of sweeping. This will create some disturbance to the floor and the disturbance to the dust that is on the floor, and then this will get suspended to the air. Dusting of different objects, slabs, almirahs, tables, windows, doors. They may remove the particle from those surface, and those particles may also be having microbial matter, and then all that microbes come to the air. And then we have seen already several times the movement of people on the chair, on the sofas, on the floor, on the bed, even in the bathroom. So these will be sort of suspending, maybe bathroom not suspending, deposited, depends on how clean the bathroom floor is, but movement of people, especially in living rooms, dining rooms, in bedrooms, study rooms, will lead to the suspension of dust and subsequent generation of also airborne microorganisms. Also now foodstuffs, especially stale food, then house plants, flower pots, pets and the bedding of the pets, sometimes furniture stuffings. They can also release fungal spores into the air. So foodstuff, maybe when it is getting stale, will lead to fungal spores, and also these other sources. In indoor buildings, an important source of proliferation and the dispersion of microbes are things that create turbulence within the indoor air. So these include air conditioners because they are throwing air into the room and basically creating air motions within the room, and that may lead to better movement of aerosols, better dispersion, and if these flows are such that resuspend the deposited dust, then also proliferation. Same for fans, coolers and humidifiers, in that they will create the air motions that will lead to a better dispersion of bioaerosols, and in some cases also an increase in the number of bioaerosol in a given unit of volume. So bioaerosol, increase in bioaerosol concentrations. As case studies or information, bioaerosols have been deposited in settings such as farms, and the main pathogenic bacteria in the atmosphere of farms, so these could be animal farms, were found to be Bacillus, Corynebacterium and Streptococcus. In museums and office buildings, other groups have been found such as Staphylococcus, Throbacter, Bacillus, Pseudomonas, Micrococcus. In hospitals, again Staphylococcus was been found, Bacillus, Corynebacterium and Micrococcus. These are some of the common bacteria that have been found. Here we have an example of the trend of bacterial and fungal count in the eye surgery ward. In the eye surgery ward, we start with the initial number, colony forming units per plate, and when the eye surgery action is happening, for some of them there is an increase during the activity, and then after the activity there is a continual increase. In terms of fungi, Cladosporium and Aspergillus, we see an increase in the count when the activity surgery is happening, and then a decrease later. So maybe these fungi were removed, but during the action, during the surgery, there were some emissions, it looks like. In an internal surgery ward, there was a slight increase in one, but in general, others did not change. Same goes for the fungi. In that Cladosporium, there was an increase after the procedure, during and after the procedure, but this was not there in the other fungi. Same was in the cesarean section ward, in that S. epidermidis increased during the surgery and then decreased after the surgery, but pretty much no change in anything else. Same goes for the fungi. In fungi, there was an increase and then a decrease in some cases, or a stabilization in another case. So in hospitals, we may be having microbes in the surgery ward, which may be due to the activities of the surgery itself. Then we have the residential apartments, where the dominant bacteria could be Aeromonas, Bacillus coccarea, Micrococcus, Nocardia, Pseudomonas, Staphylococcus. Spelling is apartments. So these bacteria can be found. In the schools also, we may find Bacillus, Corynebacteria, Micrococcus and Staphylococcus. So a wide variety of bacteria are possible to be found in the school environment. This is a case study of bacterial colonies, colony forming units in a school in Rajasthan in India, and this is the month of the year. In general, we see that the colony forming units always remain more than 200 coloniform units per meter cube of air. The lower values were in April, May, June and March, and the higher values were around September, October, December, rising up to 250 to 350 coloniform units per meter cube. So we see anyways in general we have bacterial loadings in the indoor air of schools, and this is okay, it is normal because we have a lot of human activity and sometimes cooking activities or food kept in the schools, and we have like lot of pupils, lot of teachers, supporting staff, administration, so there will be bacterial load. In another place, bacteria were studied in classroom, laboratory and seminar halls, and it was shown that the average interior bacterial levels in the classroom, laboratory and seminar halls were greater than the outside average bacteria count, suggesting that again human activities are contributing some germs or microbes to the indoor air. Also, it was seen that the doors were closed or not open all the time, which prevents the ventilation, and we know that in the absence of ventilation or at low ventilation there will be a build-up of whatever was emitted indoors. So therefore we see an increase in the indoor bacterial levels. So that is again, and due to more availability of microbes in the lavatory area. So we have already covered that lavatory especially can have more microbes. There are a lot of fungi that have been also detected in schools, from Aspergillus niger, Aspergillus fumigatus and several others, Penicillium, Chrysogenum, Aspergillus japonicus, Aspergillus ustus. So a lot of fungi are also been detected, and the main ones were Aspergillus niger, Aspergillus fumigatus and Cladosporium species. So these are there. Also these have been found in metro railway station areas, Aspergillus flavus, Aspergillus niger and the Penicillium spp. In air conditioning units in operating theatres also fungi has been found, and again Aspergillus will come, then Fusarium, Penicillium. In hospitals also the same observations has been found. Now this is a separate, so Aspergillus flavus, niger and versicolor, and this is another one, so Cladosporium will be there, Alternaria, Fusarium oxalicum, Penicillium citrinum. In toilets also fungi will be emitted, and this will depend on, of course, the type of toilet and whether the toilet lid is open or closed, and of course the extent of flushing turbulence created. It has been found that in some studies the particles produced from toilet flushes were less than 2 to 3 micrometer, even as low as 0.3 micrometer, which are the size ranges which remain suspended in the air for substantial amount of time. So a lot of the microbes that would be suspended and are attached to these particles, or are the nuclei of these particles, or they are microbes themselves and are in this size range, they will remain suspended in air for a long time, suggesting longer term exposure to anyone who will be in that washroom. So in summary, in this lecture we have seen an overview of sources of bioaerosols in different indoor environments. Can be humans, pets, schools and the washrooms.